Well, hey again, everyone. It's Rob Ryder on Saturday, September 9th, 2023. And something a little different today. Let's trust the science. Eradicating the bacteria causing Lyme disease and others, because there are all many diseases caused by bacteria, and this seems to apply to many types of bacteria that cause many types of diseases. But this will be a reading from or of eradication of Borrelia burgdorferi in vitro using chlorine dioxide, a novel approach, which was published in a European science journal. So, hey, I'm going to trust the science. On Saturday, September 9th, 2023, I am Staff Sergeant Robert Allen Ratluski, U.S. Army, veteran. But they never discharged me, so I'm still in. Also known as Rob Ryder. Email address is courtofrecord@aol.com. And find me on YouTube at Rob Ryder, ROBBBRYDER. Yes, with 3 B's. Okay, so what's this all about? Well, I had reason to go do a little researching on the internet because my son's dog apparently has Lyme disease. So I said, well, let's go see what this talks about Lyme disease online, and I found this from the European Society of Medicine. It's called the eradication of Borrelia burgdorferi in vitro using chlorine dioxide, a novel approach. And by gosh, they'll even let you download a copy of it. It's open source. So I did, and now I am going to read it to you. At least parts of it. I won't read the whole thing. But, yeah, so what did this have to tell us? Well, apparently Lyme disease, which is caused by the, I don't know if you can hear that airplane going over, but it's awful loud. Hang on a second. Okay, continuing on. Lyme disease, which is caused by the spirochete, I'm just going to call it bacteria from now on, is the most prevalent tick-borne illness in the world today and has grown into a major public health problem during the last decade. Irrelevant of decades of efforts from various health professionals. For years and years and years, they've been trying to do something about it. Ain't ever been able to figure it out. And a little bit too difficult for most scientists, apparently. The conventional treatment of Lyme disease is the use of a variety of antibiotics, but relapse often occurs when antibiotics are discontinued. And that's probably, a fact. There are several reasons why this relapse may occur, given that the bacteria is a polymorphic microorganism that can convert from vegetative spirochetes, spiral little things, to a variety of different round bodies and biofilm colonies. Well, ain't that something? Them little buggers can do lots of little things. They can change their form. Maybe that's why they're so hard to get rid of. Therefore, there is an urgent need for a novel approach that can eliminate all these different morphologies. So the damn little thing can morph into a round body, apparently, or be a spirochete, or turn into a biofilm and live inside of it. That's something. This has challenged many health practitioners around the world, not to mention the suffering of many afflicted people, millions of them. In this study, chlorine dioxide at different concentrations was tested for its effectiveness in vitro against the bacteria using combined fluorescent and dark field microscopy with liver dye staining methods. I don't know anything about it. Our experiments demonstrated that it is possible to completely eradicate all forms of this bacteria at specific concentrations of chlorine dioxide. Can we read that again? Our experiments demonstrated that it is possible to completely eradicate all forms of the bacteria that causes Lyme disease at specific concentrations of chlorine dioxide. Our extensive research has shown that chlorine dioxide can be used for the eradication of the different morphed types of bacteria, be they round bodies, biofilm, spiral thingamajiggers, whatever the hell they are. At certain concentrations of chlorine dioxide above 2 parts per million, the bacteria morphologies appear to be eradicated, as there is no motility of either spirochetes or round bodies. Only biofilms visible. However, incubating again for another 7 days resulted in the bacteria being motile, which moves, that means movement, I guess, again as they come out of the biofilms. Well, ain't that something? They hide in the biofilm until the coast is clear, and then they come back out. So apparently, you got to do something with the biofilm. We therefore decided to conduct each experiment with what we termed a regrowth kill test by incubating the initial sample with the chlorine dioxide in an incubator at 37 degrees centigrade, and the can't be back for 7 days. It appeared that the biofilms that are quickly formed as soon as the CD, that'd be the chlorine dioxide, is added in the initial experiment, broke up and released small spirochetes, baby little spirals, and round bodies of different morphologies. Well, you thought you were done, but no, they were just hiding. After numerous RKT experiments, it was determined that the concentration that resulted in near complete disinfection of the little spirally dudes as well as the round bodies was 30 parts per million CD, chlorine dioxide. Okay, so what did we learn? Well, 2 parts per million, you can pretty much eradicate so that the spirally things and the round bodies are dead. The problem is the biofilm. Got to get rid of the biofilm. So if you kick it up to 30 parts per million, apparently then it can attack what's inside the biofilm also. And when you're done, there's nothing left. So Lyme disease, also known as whatever it is caused different genome species of bacteria and most reported tick-borne infection of Europe and North America, which is also endemic in many areas in Asia. These bacteria consist of blah, blah, blah. So that's about what they are, what they look like. According to insurance claims data, 2010, 2018, the CDC recently estimated there's almost 476,000 diagnosed cases of Lyme disease every year in the United States. As the most prevalent vector-borne disease in the Northern Hemisphere, it is now considered an escalating health threat that is costing well over $1 billion per year in the U.S. alone. Well, of course, there's money to be made with them little bacteria. The general medical treatment for Lyme disease is the use of a cocktail of antibiotics for 2 to 4 weeks or longer. However, studies shown that a third to 2/3 of the patients treated with these antibiotics will suffer from persistent symptoms, a condition that has been termed post-treatment Lyme disease, characterized by infection-induced immune dysfunction or autoimmunity, chronic inflammation, persistent bacteria infections, neurological symptoms, and other tick-borne symptoms. Or, hey, maybe it was the damn medicine you gave them. Could that have been caused by the antibiotics? Well, of course it could. That's why, it's a long list of side effects. The purpose of this vitro research is to find the correct dosage of a natural antimicrobial chlorine dioxide that can eradicate bacteria in its different stages and biofilms. Be they little spirally things, little round things, or stuck in the snot. Whatever it is, going to kill the damn things. Post-treatment Lyme disease. Patients are generally treated with more antibiotics over a prolonged period, but this approach may be both dangerous and ineffective. Golly, I wonder if that could be the case. Man, if you got Lyme disease, I hope you're paying attention. Right? You need to get off that shit they got you on and go use some chlorine dioxide. The general conclusion of these studies is additional antibiotics did not benefit patients compared to a placebo group. You're as sick as you're going to be, and we can't do anything for you. We got you as sick as we can with the treatments we gave you. Right? Because really, they just want to manage your illness. They never want to cure it. They just want to manage your illnesses. Okay, the question that causes PTLD is a long-standing one. One feasible explanation has to do with the fact that bacteria, as it can change its morphology from something from the spirally things to round bodies, consists of cell wall deficient forms, L forms, or God knows, all sorts of things it can turn into, it sounds like. Changes in the internal menu with the prolonged antibiotic use can interfere with the cell wall synthesis, leading to a considerable loss of some kind of cell wall, leading to something else, which causes something else, which, or additionally, bacteria can also aggregate biofilms, which is really what's happening, right? The bacteria going into the biofilm, and antibiotics can't get through the snot. That simple. And so they just wait till it's gone. They come back out and get back at their business. So I got to read this whole thing. Hostile environments such as pH, temperature, oxidative stress, change in media composition, acidity, alkalinity, concentration of salt, sugars, other organic compounds, or antibiotic treatments can all change the morphology of them little spirally things and round bodies, or two round bodies, such as granule, cyst. So these are the different kinds of round bodies. It's nothing. They just split into, all sorts of things. Many researchers have mistaken the transformation of motin forms into round bodies as a decrease in pathogen viability. However, even though RBs, round bodies, are less motile, movable, than spirochetes, they are still able to revert to the active growing reproductive spirochetes when the conditions are favorable. When the danger's gone away, they'll come back out and change their form. Antibiotic treatment of these buggers results in some biphasic killing. Certain bacteria are eliminated, but concomitantly, another group of drug-tolerant cells appears, known as persisters. As the concentration of antibiotic reaches a certain threshold, only persister cells survive. Persisters have not been genetically modified to develop resistance. They are simply tolerant to the antibiotics in a non-inheritable way. Persisters can also transform to reverters, which is the replicating form, under the right conditions, when the coast is clear, and will therefore cause further relapses of chronic infection. Got it. The ones that survive are the ones that thrive. They're the ones that were already not affected by the antibiotic. Apparently, it's a non-inheritable way, right? There's a certain amount of them that just aren't going to be, or that will develop a resistance. Given the protective mechanisms involved in the phleomorphic abilities of the buggers in this in vitro, which I guess is test tube research, is oriented to finding effective natural substances that can eradicate all forms, including the persisters and the reverters. Get rid of all of them. This research chose chlorine dioxide for a variety of reasons. ClO2 is a small molecule with a molecular weight to something and forms a stable radical. It is smaller than the smallest viruses, so can penetrate all areas of the body, including the blood-brain barrier. ClO2 is an oxidizer, which reduced to chloride ion by capturing electron. It does so no harm to the human microbiome, as redox potential is relatively high. Now, my understanding is it's lower than what good tissue is, so you can't discharge from a lower voltage to a higher voltage. You have to go from a higher voltage to a lower voltage. So this is 0.95 volts, and most bacteria are less than that, so it can discharge into them instead of vice versa. Another reason to choose ClO2 is its ability to remove biofilms swiftly because it is highly soluble in water and can penetrate biofilms rapidly to reach and kill microbes living in the film. And that's the rest of the story. It can get the stuff that's in the biofilm. Most research on these species has been focused mainly with antibiotics. Well, go figure. However, very little research has been published about their susceptibility to disinfection using natural products such as chlorine dioxide. So when Trump back in the day was talking about, he was talking to Scarf Lady at the podium that one day about injecting disinfectants, and her eyes got really big and she about fainted. They'd make you sound like, oh, you can't do that. But apparently the science says you can, as we'll see. Chlorine dioxide is a bactericidal, virucidal, sporadical, cystical. It's got all sorts of things. And fungicidal, angicidal, cysticidal, sporicidal. Several researchers and different research studies have shown that chlorine dioxide is a strong oxidant that can greatly inhibit and eradicate microorganisms out of different concentrations ranging from 1 to 100 parts per million. Indeed, it was shown that chlorine dioxide has a potent antiviral activity in activating 99.99% of viruses upon a 15-second exposure. Just got to give the stuff 15 seconds to work, people. Find a way to make it happen. One of the remarkable properties of chlorine dioxide, it has not been achieved by either allopathic or natural medicine, is the breakdown of the protective biofilms due to their hydrophilic properties. Unlike ozone, chlorine dioxide does not react with the extracellular polysaccharides of the biofilm. Therefore, it is capable of penetrating biofilms rapidly to search and kill the microbes living within the film. Reach and kill. All right, search, reach, and kill. Get rid of the damn things. There are many reports ClO2, that'd be chlorine dioxide, has virucidal activity. The inactivation concentrations against various viruses is 1 to 2 parts per million in polovirus, 2.19 parts per million in coronavirus, 7.5 parts per million in hepatitis A. All that shit could be gotten rid of at a concentration of 8 parts per million. If you had any of those, you'd get rid of all of them. Oh, and 0.2 parts per million for rotavirus. One more. Right, very low concentrations. Chlorine dioxide has been used for over 50 years and is shown to be non-toxic, safe, and with no adverse effects on health. Well, there's the science for you. Don't argue with the science. Chlorine dioxide has been used in areas such as water treatment, textile industries, bleaching, pulp and paper, and elimination of unwanted tastes and odors. Well, those will be caused by bacteria. Various toxicological tests have been tried over decades, such as drinking chlorine dioxide, do it quite often, dissolved in water, injecting it directly into the blood under the skin of animals. Haven't tried that yet. And into the brains of mice, adding it to tissue cultures as well as using it with burns, disinfecting seeds, insect eggs, and injecting into stalks of plants. One study with human volunteers had shown that drinking chlorine dioxide up to 24 parts per million of water demonstrated no adverse effects, not a bit. Moreover, there was no evidence found of fetal malformations or birth defects with chlorine dioxide concentrations up to 100 parts per million in reproductive toxicology or tetralogy or teratology. Yada, yada, yada. Got all sorts of tests. Okay, so what did they do? Well, they went and bought chlorine dioxide, 3 different forms. CD's S+, which is a patented pure buffered solution of chlorine dioxide. I want to get some of that stuff. It's got a pH of 7. That's what you could inject into your body. You just got to... Right, because really the best way... Now this is just me talking. It isn't in here, right? This is me talking. You got to get it in the bloodstream. If you want to get it all through the body, you can't just put it on one spot of your body, or you can't drink it, ingest it. You need to get it into your blood. And the best way to do that would be with some form of saline drip with the chlorine dioxide just going in. Get the percentage in your body up to whatever it needs to be, how many parts per million, and you will kill off all these things at one time, and right, they'll all be gone. Yeah, and this is all about the test, right? So preliminary experience found the CD acts, chlorine dioxide acts on these bacteria immediately, making all the forms immotile within seconds. So measuring contact time, so superfluous. If you can get it to it, it will kill it immediately. However, if the non-moving sample was left for a day or so in an incubator and re-examined or on a microscope, small round bodies would regain their motility again. It was therefore decided to add what we termed regrowth kill tests, testing the sample with added CD again 7 days after the initial viewing. So after exposure to the CD, the bacteria were washed. So they redid their experiment. 1 week post-inoculation with the CD, the cells were again removed and re-examined under dark field microscope using some filter. These experiments were run in triplicate, and the average was taken. That bacteria was observed moving spirochetes and round bodies, as well as fluorescence dead cells. Eliminated counts were made, and each of the concentration CD used, yada, 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 blah, blah, blah. Then they got pictures and so forth, and that's all about it. But, let's see. So let's just look at some of their data now. All right, 2 parts per million. You got 1 part per million, 80% we're still alive. But at 2 parts per million or higher, 0% motility, which I believe is movement. Right, but after the 7 days with the regrowth, they were coming back. Not 100%, but like 60%. So why did I highlight these? The initial pilot experiments are found that samples that had been disinfected with 2 parts per million or higher and showed 0 motility if they were placed back in the incubator for a few days, and round bodies would come out of the biofilms and would be motile again. So you have to give it enough time to get into the biofilm. That's all this is saying. Right? As soon as it gets through the biofilm and gets to it, it'll kill them. And so you have to get your concentration up high enough, long enough for it to do that. It appeared that the biofilms that are quickly formed as soon as the CD is added in the initial experiment broke up and released their small pyrochetes around bodies of different morphologies. So when 2 parts per million CD is initially added, biofilms were immediately formed and there was 0 motility detected. However, 7 days after, they came back. So kick up the parts per million for a little bit longer, and because they're putting this in one time and then they're done. So why don't you just have it in for like a day, for 24 hours? There was one concentration of chlorine dioxide, that'd be about 30 parts per million, that decreased the mobility to 2% during the RKT. These were the only round bodies with no spirochetes in any size visible. These were only round bodies. This experiment was repeated in triplicate, yada, yada, yada. So again, they got it down to 2%, but they didn't get it down to 0 because they didn't continue it long enough to, every time you introduce the CD, it's going to cause biofilm to be created. And you're going to have to give it long enough time to get through the biofilm. These experiments have brought some interesting results to light that hopefully will enrich research literature and help clinicians treating Lyme disease to make better clinical decisions. Or you could tell them. It appears that chlorine dioxide is a powerful antimicrobial that will help to eradicate these bacteria. However, our experiments have shown that it appears to be a dose-dependent, works best at concentrations of 30 parts per million. The reason's not clear. In the initial experiments, even dosages of 2 parts per million or higher were seen to decrease the motility of the spirochetes around bodies to 0%. It's that damn biofilm they have to deal with still. The near complete disinfection was attained at 30 parts per million. Chlorine dioxide for 7 days. Yeah, we got that. So it's interesting to note that the research has found 30 parts per million chlorine dioxide was the optimal level required in vitro to eliminate motility in the initial experiments, as well as regrowth kill tests 7 days later. Clinical trials need to be completed to gain clinical experiments and what would work best. However, extrapolating from the in vitro research, it would be reasonable to use the following dosages. They're going to tell you what to do. For the average 70 kg adult, it would require 1 ampule of the CDS pure they talked about earlier in 500 milliliters of saline. It'd be a 1 to 100 dilution. Alternatively, 20 milliliters of CDS plus in a 1 liter bottle of water, drunk over a few hours. It is likely that it would need to be repeated a couple of times per week until the level of Borrelia is eradicated without giving the opportunity to regrow. Got to keep the percentages up. Various clinicians treating Lyme disease have postulated that the round bodies as well as the spirochetes can penetrate muscles and organs and hide there, protected from any type of antimicrobial. The chlorine dioxide molecule, however, is so small that it is smaller than the smallest virus and can therefore penetrate through the blood-brain barrier and other tissues of the body. Hope that the author, clinicians, the researchers, the scientists we're talking about will take these results and build on them. Okay, well, there's the Reader's Digest version of the science. I wanted to add one other thing. That there's a gentleman online, his name is Andreas Klacker. That's the easiest way to find it. Andreas Kalker, Kalker, not Klaker, Kalker. He's a German researcher, right? He's done a whole bunch of different work on this. He has these clinical studies with chlorine dioxide that you can download. But he also has all these protocols to use for whatever ails you, and using chlorine dioxide as the natural remedy for the condition that you have. And he has these videos here, and although they are in German, if I'm not mistaken, got to go here, got to go here. Hang on here. He has this one particular video where he shows red blood cells right here. And So he shows red blood cells, and if you put chlorine in, what happens to them, and then something else, what happens to them. But when you put chlorine dioxide in, what happens to them? And all these red blood cells, see, they're all clumped together, and they're nasty looking. But they all get oxygenated by the addition of chlorine dioxide, and it restores their health. So I don't think I can show you this because it'd be some kind of problem with YouTube. So I'm not gonna do that. I'm just saying you can go there and find it for yourself, and you can watch it. He puts it in. You can see what happens to the cells after he does it, and quite interesting. Okay, that's all I had to say. Right? It's short and sweet. We're talking about the European Society of Medicine. We're a bunch of scientists saying what the science on Lyme disease is. And so let's trust the science, eradicating bacteria causing Lyme disease and others, as we saw. Coronavirus. Who would have thought? Oh, isn't that what, what's going to be a deal with again here real soon? Is another coronavirus outbreak? Okay. Well, have a great day. We'll see you now.